Okay, so welcome everyone again to the Green Ear Hub webinar series. My name is Barry Crosby, and I'm one of the communication officers for the Green Ear Hub project. If you've been here with us before, you'll know that the Green Ear Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERANETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here in October last year, and we've had four very successful series so far. The first one was circularity in agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was sustainable food systems. And the one that we just finished was precision agriculture technologies for a sustainable future. If you're interested in listening to the talks related to any of the topics from the series just mentioned, please do be aware that all recordings from the Green Era Hub Breakfast Club are now available to view on our dedicated YouTube channel. Now, for the next 3 months, we're focusing in on the topic of plant health and protection, where we'll be hosting a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. And just a few housekeeping announcements. Please do feel free to type your questions in the Q&A and chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have questions for either of our speakers, please do remember to address the speaker directly in the question. Now, today we have two panelists joining us, Dr. Marika Samara and Gudrun Strauss, and we'll open our session this morning with Dr. Samara, currently a senior research scientist at Tallinn University of Technology in Estonia, where she specializes in plant virology. With extensive experience as a researcher and guest scientist at institutions across Europe, Dr. Samara's work has significantly contributed to our understanding of the diagnosis and epidemiology of viruses affecting cereal crops. Her research interests span biosciences, microbiology, and agricultural sciences, with a particular focus on viral infections in plants and their interactions with pests. She has received several awards for her contributions to the field, including recognition as the UFresco Scientist of the Month in 2024. Dr. Samara is also involved in various international committees related to virus taxonomy and plant health, through which her work continues to influence agricultural practices and research in plant virology and crop protection. Today, Dr. Samara's talk is entitled Diagnosis and Epidemiology of Viruses Infecting Cereal Crops, which is part of the UFresco Phytosanitary Research Initiative. Dr. Samara, thank you very much for accepting our invitation for our webinar series. I'm going to stop sharing my screen now, so you can start sharing your presentation. Thank you very much.
So, good morning, everybody. Thank you for taking part of this seminar. And yeah, I think I will start. So, my career in science started as a plant virologist, studying viruses, particularly Coxsackie mottle virus. And it expanded to the full genera to study also the others, because many of these viruses were biologically characterized but not sequenced. And when sequencing these different viruses, we were in one moment shifting to the high throughput sequencing surveys, and also realized that it would be really good also for surveys in the fields. So then we switched to the cereal virus field surveys. And now And now we are also thinking about, we were discovering many different viruses, and now we are thinking how to prevent these infections. And we are starting to move towards the studies on microbiome role in the infections. So beamoviruses. Perhaps many of you have never heard about those because it's a small genus of viruses that involves viruses with narrow host ranges. These viruses are transmitted mainly mechanically, but also by several insect vectors. They were in the focus of the studies first as the models for particle architecture, which was first resolved for the southern bean mosaic virus, which was the type member of this genus, and it was also giving a name to that genus. And another interesting aspect of that genus is that their genomes are very small, but they are using really various gene expression strategies, which include leaky scanning, ribosomal frame shifting using subgenomic RNAs, and also using alternative start codon. The best known virus in this group is rice yellow mottle virus. That is the most devastating virus in rice in Africa. It is mainly carried to the plants from wild rice reservoir plants by the beetles, and then it spreads via the contact transmission between the plants and also by the water and different chewing insects, animals, and birds. Now, this genus belongs into the family Solimaviridae, which also includes Inamo-virus and Cholera-virus, which were previously considered as the members of the family Luteviridae. And also a small genus Polymovirus that is represented only actually by one virus species, which is a natural recombinant between Sabimovirus and Cholera virus. And most recently, maybe also most interestingly, we have discovered that actually there are also fungal viruses that share the sequence similarity with the other viruses from Solimaviridae, and and these sequence similarities are mainly found in the open reading frames encoding serine proteases, VPG proteins, and RNA-dependent RNA polymerases, which are the new the new sorry, which are the new new characteristic which is used for RNA plus RNA virus phylogeny lineages. And also the code protein of this virus is sharing some similarities with Sabimoviruses. here you can see the phylogenetic tree that also includes this new genus, Hübschlerovirus. The coat protein genes from Enama and polariviruses are different. Also, the transmission pathways of these viruses are different because those are transmitted by the aphids. And this is determined by the open rating 3.5 product. So why do you study cereal crops? In Estonia, cereal crops are covering about 50% of the arable land. And the main cultures we grow are wheat and barley. And actually, the similar situation is characteristic also in Europe, where cereal crops occupy more than half of the arable lands. And these crops are very important in human diet and animal feed. Therefore, there are some food security concerns related to the yield reductions, which can be caused by the extreme weathers, poor soil, ineffective management practices, or damage caused by biological factors like pests, microbes, or viruses. And perhaps it's interesting to know that viruses are actually making 50% of emerging and re-emerging plant diseases globally. So they are important. And the key questions related to virus diseases are: which is the source of the virus infection? What are the hosts? How wide is their host range? And what is the disease severity caused by viral infections? Because not all viruses are with catastrophic consequences. When we were shifting to the cereal virus surveys from the very fundamental science, let's say, then we had to choose how we will approach them. The regular traditional methods were involving enzyme-linked immunosorbent assays, which were basically the serological methods, then detection using PCR, which targets the specific sequences, amplifies the specific sequences with specific primers, and also characterization of viruses using microscopical methods. But at that time, it was around 2011 or 12. Also, there was a first report of using high-throughput sequencing as a method to characterize viruses in samples without a priori information. So it is something that enables us to look for the new things which have not yet characterized, and also viruses that may maybe not seen by the traditional methods because they have mutated enough. So this high-throughput sequencing was at first mainly performed as metagenomic library construction that was based on sequencing small RNAs or RNA in the samples. And later on, there were appearing the protocols which were also approaching the amplicon specific HTS to detect specific groups of viruses. As I said, we can characterize something that is not known yet, but the restriction to that is that viruses lack universal genes. And if there are no sequencings of their relatives in the databases, then it's much more difficult to find them. Here is a little glimpse to the virus diversity. As you can see, viruses can have very different genomes. And according to this property, they were classified to the Baltimore classes. But now the taxonomy has shifted to use the signature proteins. And according to these proteins, the viruses are divided to the realms. And for example, for RNA viruses, this signature is RNA-dependent RNA polymerase. So it's kind of universal gene, but not completely, because some others don't have this gene. And for them, we will use different signatures. Among plants or plant viruses, majority of them are RNA with RNA genome. And but there are also the other groups. And this is the reason why we are widely using RNA-seq or small RNA-seq, because all viruses have to express proteins during their life. So they have to transcribe mRNA. And detection of this mRNA or the small RNAs generated during the defense process is the basis of high-throughput sequencing for plant viruses. So this is how my protocol looked like, and it can be kind of generalized. It means that first you need to extract RNA, then you have to barcode it for library preparation, to quantify it and to multiplex it equally. Then you need to choose the best platform for sequencing. Then you need to calculate how many reads you need per library. And then you have to consider the quality of the sequencing reactions. And after the quality trimming, there is lots of bioinformatic work, which includes both mapping to the known sequencing, but also de novo quantic assembly using different programs and algorithms. And after that, when you have identified your virus using BLAST against the databases, then you can start to find which are the relatives and which are the place of this new virus in phylogenetic tree, indicating its evolution. And this is for the basic knowledge But then, of course, other things you need to do, you have to figure out, does this virus needs a diagnostic assay? So it's possible now to design the primers. And also, if this virus has never characterized, it needs also biological characterization, which means inoculations and transmission assays, and also the host strains tests. In our survey, we found quite a big variety of different viruses. Lots of them were belonging to the yellow dwarf virus groups. And one of those species was new for the world. Also, we found some other viruses which were seen very rarely before, or which were seen decades ago, but which were not sequenced earlier. So they didn't have any assays for regular detection. Among them, yellow dwarf viruses were the most important ones. And important means that they are important worldwide in big growing region, as their infection can reach up with yield losses up to 86%. They are causing yellowing and dwarfing of the plants, and they are transmitted by the aphids. This is what we saw. We were sequencing around 20 isolates, which were at that moment 20% of all full-length genome isolates sent into the gene bank. And during these studies, we realized that actually the antibodies which are raised against different barley yellow dwarf species are cross-reacting with different species, and they are not reliable for the accurate epidemiological studies. Also, the multiplex RT-PCR tests which were available were really designed to discriminate species characteristic for the specific geographic region of sampling. So we got some weird results in multiplexes, and then we were also doing the in silico testing of different primers found in the literature. And we saw that even in case of 0 mismatches allowed in 3' end of the genome, many of these primers, which are shown here, were also reacting with another species. And in case of more mismatches allowed, this ambiguity raised. So for our own detection assays, we were developing a multiplex, which can be used only maybe by us, because these are the viruses which are spread here. But at the same time, we hope it's also possible to use for the others, because here you can see all the known isolates of barley yellow dwarf viruses. And these primers we designed, they actually recognize very specific sequences there. So the multiplex was fine for us. Whether it's fine for the others, I don't know. In Ireland, it was working, I know. And regarding the other viruses, there were also some interesting findings for European wheat striate mosaic virus, which is now found from the Nordic countries and also earlier from Central Europe. We found that it is adenovirus, as it was suggested, and it belongs into the Phenoi-Viridae. And what is interesting that as it has different segments, it can assort. And in Nicotiana pentamiona, actually, it will lose one of its segments. So maybe not so interesting for the epidemiological studies, but for the fundamental science, it was quite interesting. Okay, I can see that the time is running out a bit, so I try to focus better. So we also saw one Clostridium, which is under the characterization at the moment because we're looking for the vector. Then we found that brown mosaic virus is actually widely spread, and it may be because of it has different transmission pathways. It can be transmitted mechanically or by insects, but also by Puccinia graminis tritici. Then we found wheat dwarf virus in wheat. This is one of the highly pathogenic viruses. Then we also found oat sterile dwarf virus, which was previously characterized in Nordic countries mainly. And this one belongs to the genus Fichivirus and is transmitted by the plant hoppers. And together with this European wheat striate, it can cause epidemics, according to the earlier studies by Czech scientists. And also we found coxswain mottle virus, that is a Sobima virus, so it's kind of making a circle in our studies. And this finding was second time from the wheat. So I think it's only possible because of using HDS, because it's usually doesn't belong into the regular assays. And this led us to the thinking that we need a more wider epidemiologic study, because many of these viruses are, as in Estonia, which is a very small country, we found many new things, and possibly it's also true for the other countries. And also the climate change is fostering, for example, insect-transmitted viruses. And the other thing that is also now influencing the situation is the prohibiting the use of neonicotinoid insect seeds, which has led to the increased use of pyrethroid sprays, for which pests are evolving resistance. And there are also some others which are really important and difficult to control. And yeah, now I am reaching to this point. It's this was leading us to the Eufresco project, and the main targets of this project were the epidemiology, to understand what is going on, which viruses are occurrent in cereal growing areas, and also development of novel diagnostic protocols. And here, many people joined, and with different interests. So we had different book packages. One of them was about diversity, but also there were groups who focused on aphid-specific transmitted viruses or hopper-transmitted viruses. Then there was a group for soil-borne transmission, seed transmission, and also contact transmission. And what were the findings? The findings about yellow dwarf viruses, very interesting, because in Europe we saw that barley yellow dwarf virus PAS was prevalent, although in Ireland it was barley yellow dwarf MAV. And it was different, but was considered earlier, because earlier the PAV was considered the prevalent one, and also the resistance breeding programs are targeting currently PAV. So I think this is one of the most important things we figured out. And another thing that came out from these epidemiological studies was that there are very new species we didn't know before. Both insect transmitted, soil-borne, and also seed-borne ones. And the studies didn't concern only Europe, but also the other countries worldwide. So in Africa, mainly the maize and sugarcane and sorghum were surveyed. Novel viruses were also found there. And in other countries, yeah, there were more. And the other part of this study included development of the diagnostics. Where can I continue? What do you think?
I'd
Yes.
say maybe just one or two more minutes should be fine.
Oh, okay.
Yeah, thank you.
For which I think one of the most interesting things was that our Irish partners from Chagask developed a droplet digital PCR assay for MAV and Pest for the discrimination and their detection from grain aphid samples. And they also developed HCS-based sequencing assay for genotyping the aphids, both for yellow dwarf viruses, but also for the knockdown resistance of aphids, which are against the pyrethroids, which is really developing fast according to their screenings, because in the beginning it was around 0%, and in the last year it was about 18%. Then the French partners and Danish partners were also developing methods for yellow dwarf viruses detection and discrimination. And our soil-borne virus group was really successful developing new protocols for detecting soil-borne viruses. For example, this isothermal loop-mediated amplification was developed for the soil-borne cellular mosaic and Divid virus. And the German partners were also developing novel RT-PCR methods that able to detect and quantify different soil-borne viruses belonging to the furovirus group. And yeah, and also we had a group of organizations from consultative group of international agriculture research who were focused on developing protocols for viruses which are spread in tropical countries. So I would like to thank all the participants of these projects and apologize for this poor overview.
Right, good. Thank you so
Within 15
much for
minutes.
that really interesting presentation. Don't worry at all. That's absolutely fine. I think what we'll do, maybe just in the interest of time, is if anyone, well, first of all, I should just say that the chat box and Q&A section is open. So if anyone in our audience would like to ask a question, please do address Dr. Samira directly in the Q&A section, because she would be able to respond to that over the next half an hour to your question. So please do feel free to type a question. Dr. Samira, thank you so much for that. Fascinating study and a wonderful overview, I think, of the dynamics of that particular research. So really, really interesting stuff. Joining us is Gudrun Strauss, who's currently a research entomologist at the Austrian Agency for Health and Food Safety, where her work focuses on investigation of invasive insect pest species, and particularly non-native insects that affect vineyards. Her interest in pest management was sparked during a beekeeping training course, which led her to investigate an introduced plant hopper pest, and from there developed an interest in the study of biological control agents and assessing the environmental risks associated with their use in Austria. Through these studies, she has built expertise in pest identification, risk assessment, distribution modeling, and monitoring of insect pests. In recent years, Gudrun has concentrated on the genus Scapoides titanus, a key leafhopper pest found in vineyards, where her work has focused on examining the potential use of certain fungi as biological control agents against that particular pest. Gudrun, sorry, the title of Gudrun's talk today is Improved Knowledge about Epidemiology and Distribution of Priority Invasive and Re-emerging Arthropod Pests in Fruit Crops and Grapevines. Gudrun, thank you very much also for accepting our invitation for our webinar series. We're absolutely delighted to have you here with us today. So without further ado, the floor is yours. I'm going to stop sharing my screen so you can start sharing yours.
Okay.
Thank you very much.
Thank you, Barry. Thank you for having me. And I would like to wish and welcome all the participants. It's really a big pleasure for me to join the Breakfast Club for the first time. Okay, so I start with share my screen. And okay, is it in full presentation mode?
It is, Gudrun, yes, thank you.
Okay, great. So, I was asked to present the UFresco Epidemics project, which I coordinated. And this UFresco project was realized within the UFresco network. And just for all those who haven't heard about UFresco, what is it? It is a network of organizations, mainly ministries of agriculture, fishery, or forestry. And the goal of this network is to coordinate to coordinate the research in the phytosanitary area. So, and this was one project, the Epidemics project, and as you can see, this project has been already finished. We started in March 21 and until February 2003. And the focus was on mainly 3 species, Aromia bungi, Popillia japonica, Haliomorpha halis, and Lycorma delicatula to a much lesser extent. So all those species have in common that they originate in Asia, and except for Lycorma delicatula, have been introduced into the EU and pose a major threat for fruit production and grape wines. So I will focus on these 3 species and yeah, some words about the partners. We had 14 institutions from 9 countries participating in the Epidemics project. And here on the slide, you can see the institutions. So FARA was participating from the UK, 2 research institutions from Belgium, INRAE from France, University of Azores, the Plant Protection Service from Northern Italy, also from Lombardy, from Slovenia, University of Ljubljana, from Serbia, University of Novi Sad, from Bulgaria, University of Forestry, and us from AGES. Yeah, well, the scope of the Epidemics project was quite broad. The overall goal of the project was to improve the knowledge about the target species. And it's a very important step in plant protection is early detection. And so this was the surveillance was the main objective in work package 2. And I will later on show you some of the activities that took place in this work package. Also very important is the correct identification of the target pests. So this was the goal to contribute for this in work package 3. And work package 4 was on potential containment and control measures for Popillia japonica, I have to say. But I'm not going to talk about this work package also because of the time. So now I would like to say some words about the target species and show you which activities were accomplished. So let's start with Aromia bungi, the red-necked longhorn beetle. It is a really impressive Cerambicide, native to East Asia, so from China, Korea, Taiwan, Vietnam, Mongolia. But interestingly enough, it has been introduced to Germany, where it was first recorded in 2011 in southern Bavaria. This is close to Austria, in a private garden on a plum tree. And it was more beetles were detected in 2016 and 2019 in the same area. And of course, as Aromia bungi is listed as a priority pest in the EU, demarcated areas were established and suspicious trees were felled. And the official status now for Aromia bungi in Germany is under eradication. The situation in Italy is more difficult. It was first recorded in 2012 in Campania, and later on in Lombardy, and in the meantime also in Lazio region and Tuscany, where more than 200 trees were infested. So in Italy, the official status for Aromia bungi is under containment. And yeah, Aromia bungi, the problem with this beetle is that they infect also healthy trees. Stressed trees also, but the problem is if they infect living trees. And you can see the damage is caused by the larvae, which bore galleries in the trunk and in the branches, and of course thereby weakening the trees. So because of this threat, Aromia was put on the list for priority pests in the EU. And so within the Epidemics project, we did an expert survey. The colleagues in Belgium and we from Austria, we used the same methods. We first selected sites that had a higher risk of invasion. So in the case of Austria, there is one very important transit route leading from Italy through Austria to Bavaria in Germany. So we selected prunus orchards along these transit routes. And we used these black cross traps, which you can see here on this images, together with a specific lure for Aromia bungi, which is a pheromone of Aromia bungi. And so the colleagues from in Belgium used the same method, and you see the distribution of the monitoring sites there. And during these 2 years of monitoring, no Aromia bungi were captured. In contrast, in northern Italy, where already an infested area exists, they deployed more traps in this infested area here. And the traps were checked once per month. And with this method and with this pheromone, 12 Aromia bungi were captured within this already infested area, within a radius of 100 meters. Beside this expert survey, it was also an objective to raise the awareness for these important pest species in the public. So new mobile phone applications were created by INRAE, for example, to involve the public. And as I've mentioned before, it's very important to correctly identify the pest species. And the colleagues from INRAE did barcoding with Aromia bungi specimens. And so the sequences are now available for you if you are interested in the EPO QBank database. And in the course of the Epidemics project, a diagnostic protocol for Aromia bungi was started. And I really like to recommend this diagnostic protocol because it contains really excellent images of Aromia bungi, but also on several lookalike species. And you will find a key showing and explaining all the morphological characters for the larvae, which of course are more difficult to identify. So this was started during this Epidemics project, and it was a cooperation between INRAE and the European Reference Laboratory for Insect and Mites. So now we move on to Pupilia japonica. You might have heard of it, the Japanese beetle. We are all very worried about this beetle. It is a scarabaeidae. native to Japan and the Far East, but it was introduced to several other locations of the world. First to the United States, where it really became invasive and a big problem in turf grass. But it was also reported from some of the Azores Islands, and in 2014, it was then reported for the first time from the mainland Europe. In Italy, from a national park, and from there it spread to the Italian-speaking part of Switzerland, to Tessin. And in the last 3 years, several specimens were also recorded north of the Alps, so in Zurich, for example, and also in Germany, Baden-Württemberg. And the last record last year was from Slovenia. So the problem with Popelia japonica is that it's really so polyphagous. More than 400 host plants are known, and you see it attacks so many different plant species. Fruit crops are affected, like Malus, Prunus, Rubus, but also grapevine, and it attacks the flowers, it attacks fruits. You can see the damage the beetles do. They feed on the tissues between the veins and skeletonize them. And yeah, you can see there's also a big damage for pastures because the larvae feed on the roots. So again, we did an expert survey with these funnel traps. You can see them here, together with the specific lure for Popelia japonica, which consisted of a floral scent and a sexual pheromone. And yeah, we had 21 monitoring sites in Austria, either in orchards or in distribution centers, garden centers, and yeah, again on important traffic routes. And fortunately, there were no captures of Popelia japonica. The situation in northern Italy, of course, is different, and the colleagues from the Plant Protection Service of Lombardy, they tested simple homemade traps to see if it's possible to follow and survey the phenology of the beetle. And so they installed these traps in the already infested zone, and the traps were controlled once per week. And here you can see the results. So yes, it was possible to find the first adults already at the end of May. And as you can see, the number increases during June and reaches a peak in July, and then the number quickly decreased. And yeah, the same was true for Popelia. It's very important to involve the public, and so another mobile app was developed for Popelia japonica, and also the phytosanitary service in northern Italy developed such a tool. Yeah, a very nice collaboration was for the molecular identification of Popelia japonica, because there are a lot of Popelia japonica beetles in the Azores Islands. So our colleague from the university sent the specimens to the colleagues at INRAE and they were able to do the sequencing and barcoding. And again, all those sequences are now available for you in the EPUQ databank. So coming to the last target species, Haliomorpha halis, also called brown marmorated stinkbug. It is a Pentotomide originating in East Asia. But as you can see from this map, it is a very successful invader in many different parts of the world. And it is a serious pest in fruit and vegetable crops in Northern America and Europe. And it's also polyphagous. And here are some pictures of adult beetles on cherries and apples. And you can see the deformation it causes by stinging and sucking on the fruits. Here you can see a nymphal stage. So within the frame of the Epidemics project, different trap types were tested. For example, the colleagues in Northern Italy, they tested a mass trapping system with insecticides and water beneath. And so this catched a lot of stink bugs. But on the other hand, the damage was still not reduced significantly in the orchards nearby. And we ourselves, we tested these two trap types, and yeah, we put the traps in orchards and also hazelnut orchards. And yeah, here you can see the traps. We use this black dead in pyramid trap, which has a collecting jar on the top, and together with a pheromone. This is very important to mention. With the it's an aggregation pheromone, and this really works very well. And then we had this black cross trap. And yeah, we were really surprised about the numbers we found in Austria. Here on this map, you can see that we only had higher numbers in urban areas. For example, in the capital of Styria, in Graz, we had the highest numbers, and also in Vienna, whereas in the more rural areas, there were no catches or the numbers were very low. So this was a rush through the Epidemics project, and I really want to thank all the partners that contributed to this project. I really enjoyed the collaboration, and I think that we really contributed to gain more knowledge on the pest species. And yeah, I want to thank you for your interest, and I will be happy to answer any question you might have. Thank you.
Gudrun, thank you so much. Another really excellent presentation. And may I just say the quality of your images was absolutely striking. Visually, very beautiful presentation. The images that you captured of the pests, I think our audience would agree, were really striking. Really brought the presentation to life for us. I was just wondering, just while people are waiting to type questions into the chat box or the Q&A function, I was just wondering about the kind of geographical spread of pests affecting these kind of fruit crops and vineyards. Have you identified any kind of particular location globally where there is a kind of a concentration of pests, or your presentation seemed to suggest that they're just kind of found everywhere? Or would I be right in saying that there is a kind of a disproportionate concentration in kind of East, Southeast Asia? Would that be correct, or just everywhere?
Okay, let me just recap. So your question is about if there is, let's say, a special spot in Asia where the pest species
Yes, well,
are more
kind of, I
concentrated,
suppose
or
more kind of globally. Like, so is there kind of an area, geographical area of the world where these pests are found in greater numbers, or the kind of epidemiological kind of concentration of them is kind of is much there's a much higher rate,
well,
or is it just kind of evenly spread globally throughout the world?
well, first of all, I want to mention at this point that, for example, it's true for Haliomorphohalis, it is not a pest species in its native area. So, and when you look where this species was brought to, yeah, uh, it it is quite obvious that this has to do with trading, yeah, or hitchhiking also. This is true for Pupilia japonica because single specimens were found, for example, then at a really great distance from yeah, other outbreak areas. So along railroads, for example. So yeah, human influence and human activity plays a major role. Yeah. Also, of course, trading of plant material. I don't
Fantastic.
know if this answered
No,
your
absolutely.
question, but yeah.
Thank you so much. Very interesting. So just a question from a member of the audience. Dear Dr. Strauss, I am Sama, a PhD student from the University of Trento, Italy. I'm working
Yeah.
on vibrational communication of stink bugs, and I think it would
Yes.
be very interesting to study the vibrations of the chewing of larvae of Aromia bungi and maybe develop a detection system based on the chewing vibrations. Could be a good strategy for prevention. Have you explored this possibility yet?
No, no, no. I think this is a very, very interesting idea. The problem with Aromia is that they attacked also healthy trees, and of course it's hard to find out if a tree is already infested or not. So it would be great to have a tool to find out which kind of larvae is inside a tree. Is it a worrying one like Aromia bungi, or is it a native species? Yeah, I think on this field of vibrational communication, and to study it as also maybe for biological control. I know this for one of my main insect pest species is a leaf hopper, Scaphoideus titanus, and I know that Italian colleagues worked a lot on vibrational communication of this species. So it's very interesting. Go ahead.
Thanks, Gudrun. Thank you. Um, so just one other question. Thank you for the talk, Gudrun. Were any ideas posited as to why Halomorpha halis was found more often in urban centers in Austria?
That's a very good question. That's a very good question. So the first thing that we were considering, maybe they find better spots to hibernate during to spend the winter in urban areas. So this could be one explanation. But we don't know. We were surprised because we were expecting higher numbers, in fact. And then we had almost no catches in the in the rural areas. Yeah, and we stopped the monitoring now because it's not the pest species in Austria, and the numbers are not worrying. Yeah, it's completely different. It's a completely different picture to, for example, northern Italy. Yes.
Excellent. Gudrun, just one quick question before we let you go. Just remember the audience. Can you comment on which trap types were most effective for trapping Papillia japonica and Aromia bungi during the project?
Well, for Aromia bungi, we tested this black cross trap together with this sexual pheromone, and this is a pheromone from this species. So it's so important. You have to use this pheromone. It's absolute necessary. And I showed this. The Italian colleagues also used this pheromone and were able to record Aromia bungi. For Papillia japonica, you can use there are different trap types. Also different color combinations. So we use the yellow and in combination with green, and this specific lure, which contains not only sexual pheromones, but also this floral scents of organole. And this is important to attract successfully the beetles. But there, we did not test different color combinations. So I really can't recommend any kind of color combination. I may add that for Haliomorphohalis, we found that these pyramid traps, which are put on the ground, catch more nymphal stages. Because the nymphs are able to climb up from the ground into the collection jar, whereas in the cross traps, which we are hung in the trees, we were not able to collect that much immature stages.
Gudrun, thank you very much. I think just
Thank
in the
you.
interest of time, I think we'll just stop it there. But I think we could go on and the same as well for Marika's talk. I think we could be here for another hour easily with questions for both of you. Two fantastic presentations and very, very interesting projects. And we wish you all the very best with those projects as the year progresses. Our next webinar will be on March the 20th, when we'll continue our webinar series on plant health and protection. I'll be sending out the invitation to all of you in the following days. In that breakfast club, we will have Dr. Chiel Pell, Scientific Officer at the Netherlands Food and Consumer Product Safety Authority, discussing taxonomy and epidemiology of Pectobacterium and Dicea species in Europe, North America, and South Africa. We'll also have Ellen Everard, researcher at the Flanders Institute of Agriculture, Fisheries and Food in Belgium, talking about detecting virus-carrying Symphonema species as an alternative to Symphonema identification up to the species level in trade. Again, thank you very much to both of our panelists today, and thank you, the audience, for participating, coming along this morning and asking questions. We really appreciate your time. Wishing everybody a fantastic weekend ahead, and hope to see you next time around at the webinar. Thank you very much.